when you are, huddled under a thick blanket with 101-degree fever, shivering, completely exhausted.
And refusing to eat anything more than, like, a saltine cracker.
Right, exactly. You usually feel like your body is just failing you, like it's a breakdown.
Oh, absolutely.
But the reality is actually the exact opposite. That miserable state isn't a malfunction. It is a flawless, billion-year-old biological program executing its code.
It really is.
Whether you have a stomach bug, a bacterial infection, or the viral flu, your symptoms are remarkably identical. And that's because your body is doing that on purpose.
Yeah, it's a highly conserved survival mechanism. I mean, the pathogens themselves aren't turning off your appetite or, cranking up your body heat.
Right.
Your own brain is pulling those physiological levers to basically create an inhospitable environment for the invader.
Which is amazing to think about.
It is. But the enduring mystery in neuroscience has always been, well, how the brain actually orchestrates this. Like, how does it know you are sick? And how does it manage to trigger all these different physiological changes simultaneously?
Which brings us to the mission of today's deep dive. We are unpacking a phenomenal piece of research published in the journal Nature. These scientists have actually found the brain's master control room.
A specific sickness hub.
Exactly, a sickness hub. So today, we're going to trace the entire pathway. We'll explore how the brain senses a threat, the cutting-edge tools they use to map the specific neurons responsible, and the physical wiring diagram that turns off your hunger and induces a fever.
And to map that pathway accurately, the researchers, they couldn't just infect mice with a random cold virus.
Because it's too unpredictable, right?
Exactly. Live pathogens are super unpredictable. They replicate at different rates. They cause varying levels of tissue damage. And that just introduces way too much noise into the data.
Right. You need a clean baseline.
Yeah, they needed a perfectly standardized sickness, so they achieved this by using two very specific chemical mimics.
So they essentially tricked the immune system without risking actual tissue damage.
Exactly the concept. They used LPS, which is lipopolysaccharide.
Okay, lipopolysaccharide.
Yeah, it's basically a structural component found on the outside of bacterial cells, so it mimics a bacterial infection. And they also used polyic, which is a synthetic molecule that mimics the double-stranded RNA of a replicating virus.
So one fake bacteria, one fake virus.
Right. And injecting either of these creates a massive immune response without a live pathogen. And the results were incredibly uniform.
Oh, really? Like exact same timing?
Practically, yeah. In both cases, the mice developed a fever that peaked exactly 2 hours after administration, right alongside lethargy and a total loss of appetite.
Wow.
Because the physiological response was virtually identical, the researchers primarily relied on the bacterial mimic, the LPS, to map the rest of the neural circuits.
Okay, so the body detects this fake bacteria, the immune system goes to war, but I'm looking at a logistical problem here for the brain.
Right, the vault.
Yeah, the brain is housed behind the blood-brain barrier. It's a highly secure, sealed vault designed specifically to keep peripheral blood and all its chaotic fluctuations out of the sensitive neural tissue.
Which is normally a very good thing.
Right. So how does the alarm bell happening in your bloodstream actually get heard inside that sealed vault?
Well, the barrier isn't uniformly impenetrable. There are highly specialized locations called circumventricular organs where the barrier is intentionally porous.
Intentionally porous, so like a window.
Think of them as tiny windows, yeah, looking out into the bloodstream. And the researchers focused on one in particular called the OVLT.
The OVLT.
Right, the vascular organ of the lamina terminalis. It's located in the preoptic area of the hypothalamus.
Okay, let me use an analogy here. The blood-brain barrier is like a massive fortress wall, but this OVLT is like a porous border checkpoint.
That's a great way to picture it.
So the immune system's alarm signals drift up to this checkpoint at the OVLT. Do they physically cross the threshold into the brain tissue?
Not exactly. The physical signals, the cytokines and inflammatory molecules produced by your immune system, they interact directly with the cells lining that border.
Oh, I see.
And that interaction sets off a chain reaction. To see where that reaction was heading, the researchers looked at the brain tissue immediately adjacent to the OVLT, a region known as the VMPO.
The ventral medial preoptic area.
You got it, the VMPO.
And what's happening in the VMPO during this simulated infection?
It lights up like a switchboard.
Really? Just the whole area?
Yeah, and they quantified this by staining for a protein called FOS.
FOS. Let's explain FOS for a second.
So FOS is an immediate early gene product. When a neuron fires rapidly and repeatedly, it produces FOS protein almost immediately. It's essentially the biological exhaust fume of a highly active cell.
Exhaust fumes. I like that. So if a cell is working hard, it's pumping out FOS.
Exactly. And in the mice injected with the LPS, the entire VMPO region was saturated with this FOS protein.
Okay, let me do a quick mini recap to make sure I'm holding all these pieces together for you listening. Bullet 1, we have a standardized bacterial mimic, LPS, triggering an immune response.
Check.
Bullet 2, the chemical alarm reaches a leaky border checkpoint in the hypothalamus called the OVLT.
Right.
And bullet 3, immediately next door, the VMPO region is churning out FOS protein, proving it's highly active.
Exactly.
Which leads to a fascinating question for you listening. Next time you feel a fever coming on, picture this tiny border checkpoint in your hypothalamus.
Right.
What kind of cells do you think are waiting on the other side to sound the alarm?
Well, finding activity in a geographical region like the VMPO is a great start, but it's kind of like saying the call is coming from inside Manhattan.
Right, it's a big place.
The brain is a dense, tangled jungle of billions of cells. We needed to find the specific trees in that jungle that were on fire.
So the researchers brought in some incredibly heavy, modern scientific artillery. 2 high-tech tools that completely change how we look at the brain.
Yes. The first is a technique called snRNA-seq, which stands for single-nucleus RNA sequencing.
Single-nucleus RNA sequencing.
Right. If you want to understand what a single cell is doing, you look at its RNA. RNA is the instruction manual the cell is actively reading to build proteins.
Okay.
By sequencing the RNA of tens of thousands of individual nuclei, we can see exactly what jobs those cells are specialized for. It's like intercepting the cellular mail to read their daily work orders.
So snRNA-seq is intercepting the mail to see what they're making. What about the second tool?
The second is MERFISH, multiplexed error-robust fluorescence in situ hybridization.
That is quite the acronym.
It really is, but its function is beautifully intuitive. While snRNA-seq tells us what the cells are doing, the process of sequencing destroys the tissue, so we lose their physical location.
Oh, so you know what the mail says, but you don't know whose house it went to.
Exactly. MERFISH solves this. It uses fluorescent probes to light up specific RNA sequences within an intact slice of brain tissue. If snRNA-seq is intercepting the mail, MERFISH is the Google Earth GPS tracker that pins the exact physical coordinates of the house receiving that mail.
I love that mnemonic. So we have the intercepted work orders and the GPS coordinates. Applying this to the VMPO region, they analyzed over 16,000 individual nuclei.
Yeah, massive amount of data.
And they grouped these cells based on their genetic mail and found 32 distinct clusters, 32 different cell types coexisting in this one tiny neighborhood.
But when they cross-referenced those 32 clusters with the FOS protein, our marker for the cells actually firing during the sickness, 2 groups stood out dramatically.
Just 2?
Just 2 main ones. The first was cluster 19, which are neurons. The second was cluster 5, which are non-neuronal support cells.
Okay, let's look at the neurons first. Because they activated in response to the LPS mimic, they named them VMPOLPS neurons.
Right.
And what did their genetic mail tell us about them?
It gave us a highly specific molecular signature. These are inhibitory neurons, meaning their job is to send a stop signal to other circuits down the line.
A stop signal. Got it.
And we know exactly how to identify them because they constantly express 3 unique genetic markers: galanin, calcar, and amigo2.
Galanin, calcar, and amigo2.
Think of that combination as a highly specific uniform. If you want to find the exact cells orchestrating your flu symptoms, you look for the cells wearing the galanin, calcar, and amigo2 uniform.
Okay, so synthesizing this with a quick recap. Bullet 1, snRNA-seq reads cell function. MERFISH maps cell location. Bullet 2, our high-tech tools isolated the exact neurons holding the master control levers, identified by that galanin, calcar, and amigo2 uniform.
Perfect.
And bullet 3, non-neuronal cells, that cluster 5, were also activated, but I'm going to push back here. I'm getting stuck.
Okay, what's the hang-up?
If neurons are the brain's wiring computing information, why were the non-neuronal cells, the structural support staff, also lighting up during an infection? I mean, they should be able to smell bacteria, right?
That is the exact paradox the researchers had to solve. Neurons are specialized for rapid electrical communication. They are largely blind to peripheral immune threats.
Right.
The immune system is the sensory organ for infection. The connection between the two relies on physical proximity and a process called paracrine signaling.
Paracrine signaling. Let's break that down.
So paracrine signaling is basically cellular whispering. It's when a cell releases a chemical message that only travels a microscopic distance to affect the cells immediately adjacent to it.
Okay.
Remember our Murphish GPS tool?
Google Earth pin.
Yeah. What's fascinating here is it revealed that 90% of those activated VMPOLPS neurons were located within a mere 200 micrometers of the base of the brain. They are packed right against that leaky OVLT border.
200 micrometers. They are practically leaning against the glass of the window.
Exactly. And sharing that exact same space are the non-neuronal cells from cluster 5, specifically endothelial cells lining the blood vessels and ependymal cells lining the fluid ventricles.
Okay, so the support staff is right there at the window too.
Yes, and these cells actually do interact with the immune system. When the peripheral alarm molecules from the blood hit these border cells, the border cells act as a neighborhood watch.
Oh, the neighborhood watch.
They detect the threat and immediately start shouting to wake up the neurons.
And their version of shouting is secreting chemicals.
The researchers found that these border cells start pumping out high volumes of cytokines and prostaglandins.
So specific chemical messengers.
Right, specifically molecules called IL-1 beta, PGE2, and CCL2.
Let me guess, those are intense inflammatory alarm bells.
Incredibly intense.
But releasing an alarm bell only works if someone has the ears to hear it.
Which brings us back to the genetic makeup of our VMPOLPS neurons. The sequencing data prove that these specific neurons are biologically hardwired to hear that exact chemical shout.
Oh, wow.
Yeah, their surfaces are studded with the precise receptors needed to detect those molecules. They have the IL-1 RAP receptor to catch the IL-1 beta. They have the EP2 receptor for PGE2 and the CCR2 receptor for CCL2.
It's a perfect lock and key system.
Exactly.
But wait, how do we know for sure that those specific chemicals actually make the neuron fire? Like, seeing the lock and key is one thing, but proving it turns.
They proved it using patch clamp electrophysiology.
Okay, explain that.
They took a microscopic glass pipette, attached it to a single VMPO-LPS neuron in a petri dish, and measured its raw electrical resting potential.
Okay, so they are physically tapped into the cell's electricity.
Right. Then they washed those alarm chemicals, the IL-1 beta, PGE2, and CCL2, over the cell. Immediately, the electrical potential shifted. The neuron became highly excitable, and its firing rate spiked.
Wow.
The lock was turned, the neuron woke up, and the master signal circuit was initiated.
Let's pause and pull this all together with a quick recap. Bullet 1, non-neuronal cells detect immune signals at the barrier. Bullet 2, they act like a neighborhood watch, secreting IL-1 beta, PGE2, and CCL2.
Yep.
Bullet 3, the VMPOLPS neurons, parts just micrometers away, have the exact receptors for these and become highly excitable.
Nailed it.
So question to you listening, does knowing that your brain's support cells act as an internal immune alarm change how you view brain fog during a cold? I mean, it's not just a vague sense of fatigue. It's active, localized chemical warfare happening in a highly specific neighborhood of your hypothalamus.
It really radically changes how you visualize being sick, but at this point in the research, we still only have correlation. We know these neurons fire when you are sick.
Right.
But to do rigorous science, you have to prove causation. Are these specific neurons actually the ones generating the physical symptoms of a fever and appetite loss?
Right. Are they the cause? And finding out required a method to control the neurons, to essentially turn them on and off on command. Here's where it gets really interesting.
Where it's wild.
They didn't just look at the cells. They used a mind-bending genetic system called TRAP2, targeted recombination in active populations.
The TRAP2 system is, it's one of the most elegant tools in modern neuroscience. It allows you to take a permanent genetic snapshot of a temporary event.
Okay, how does that work?
Here's how it works. They use genetically modified mice where a specialized enzyme, recombinase, is tethered to the FOS gene.
Okay, let's track the logic here.
Yeah.
FOS is the exhaust fume. It only gets made when a neuron is firing.
Right.
So if the enzyme is tethered to FOS, the cell only produces the enzyme when it's actively firing.
Precisely. So they inject the mouse with the bacterial mimic. The mouse gets sick, and the VMPOLPS neurons start firing and producing FOS, which means they're also producing this special enzyme.
Okay, got it.
Right at that exact moment, the researchers inject a drug called 4-OHT. This drug activates the enzyme, and the enzyme permanently installs a synthetic receptor called HM3D directly into the DNA of those active cells.
Wow. It's like pouring genetic amber over the circuit while it's running.
Yeah.
The surrounding neurons that aren't involved in the sickness response, they don't have FOS.
Exactly.
So they don't get the synthetic receptor. You've essentially installed an exclusive on-off switch only into the sickness hub.
That's the beauty of it. So first, they test for sufficiency, meaning if we manually flip the switch, can we cause a fever without any infection present at all?
Okay, testing sufficiency, turning them on in.
The synthetic HM3D receptor they installed does absolutely nothing on its own, but it responds to a designer drug called CNO. So they took healthy, uninfected mice who had previously been trapped and gave them CNO.
So the CNO binds to the synthetic receptor, forcing the sickness hub to fire, even though there's no bacterial mimic, no immune response, no alarm bells.
Correct.
What happened?
The mice immediately exhibited profound sickness behaviors. They developed a significant fever. They displayed warmth-seeking behavior, actively moving to a zone in their enclosure that was 2 degrees Celsius warmer than their normal preference.
Yeah.
And their appetite completely plummeted. They ate 60% less food. We forced the neurons to fire, and the symptoms manifested instantly.
That definitively proves sufficiency. Those neurons alone are enough to cause the symptoms. But what about necessity?
Right, turning them all off.
Yeah, if we delete those neurons, does the body lose the ability to mount a fever during an actual infection?
To test that, they used the TRAP system again, but instead of installing a switch, they installed the genetic instructions for diphtheria toxin. This effectively ablates or destroys just those sickness-activated neurons.
So they physically removed the sickness hub from the circuit board.
Yes.
And when they gave those mice the bacterial mimic,
The mice did not get a fever, their core temperature remained completely flat, and they did not seek out warmer environments.
amazing.
Yeah, the peripheral immune system was still reacting to the mimic, but the brain's command center was gone, so the physiological levers couldn't be pulled.
I do want to mention, looking at the data, the appetite wasn't completely restored in the ablation experiment. They still lost some desire to eat.
That's a crucial detail. Biology hates a single point of failure. The researchers hypothesized that while this hub is the primary driver, suppressing appetite is so vital for conserving energy during an infection that there are likely parallel redundant circuits also contributing.
That makes sense.
But for fever and warmth seeking, the VMPO-LPS neurons are absolutely necessary.
Let's synthesize this incredible experiment with a mini recap. Bullet 1, TRAP2 allows researchers to tag and manipulate only the infection-activated neurons.
Yep.
Bullet 2, activating them causes fever and appetite loss without an infection. Bullet 3, destroying them prevents fever even when an infection is present.
Spot on.
So question to you listening, if we can literally turn a fever on and off like a light switch, what does that mean for how we treat severe fevers in the future? I mean, we wouldn't need broad-spectrum anti-inflammatories. We could just quiet down the specific circuit.
It really is the ultimate precision medicine. But, knowing the command center is only half the battle.
Right.
How do neurons sitting in the preoptic area of the hypothalamus actually reach out and physically alter the temperature of your entire body or stop your stomach from craving food?
They had to trace the wiring diagram. Where do the physical cables, the axons from the sickness hub, actually go? And to figure this out, they used a technique involving a modified rabies virus, which sounds totally counterintuitive. Why use a deadly pathogen to trace healthy brain circuits?
It does sound wild, but standard chemical tracers just diffuse everywhere. They don't respect the synaptic boundaries between cells.
They just bleed over.
Exactly. Rabies is entirely unique. It has evolved to specifically infect neurons and travel backwards, retrogradely across the synapses, jumping from the receiving cell back to the sending cell.
Oh, that's clever.
Right. By using a genetically modified safe version of rabies that can only jump exactly one synapse backwards, researchers can inject it into a downstream target and watch it travel back to light up the exact upstream controllers.
It perfectly traces the chain of command. So let's trace the wires. Where do they go to control the fever and heat the body up?
The rabies tracer revealed dense projections from the sickness hub to 2 specific areas downstream, the AVP and the MNPO.
Hold on. I recognize those regions. Those areas house the brain's primary warm, sensitive neurons.
Yes, they do.
Those are the cells that constantly monitor your blood temperature. And if you get too hot, they trigger your body's natural cooling systems. They make you sweat, dilate your blood vessels, make you seek shade. So to heat the body up, the sickness hub is sending signals into the cooling center.
Yes. And recall the specific genetic signature we discussed earlier. The VMPO-LPS neurons are inhibitory.
Oh, Wait, that makes perfect sense. So they are sending an inhibitory signal to shut down the cooling system.
Exactly.
It's like turning off the AC to heat up the house. If you disable the cooling mechanism, the ambient heat naturally rises, creating a fever.
It's so efficient. And they verified this using optogenetics.
Okay, light-sensitive proteins.
Right. They inserted light-sensitive proteins specifically into the axons connecting the sickness hub to the cooling centers. When they pulsed a laser to manually activate that inhibitory connection, the cooling center shut down, and the mouse's core body temperature spiked.
Disabling the biological AC to create a fever. That is remarkably elegant. What about controlling the appetite? How do they stop you from eating?
For appetite, the rabies tracing showed direct connections to the arcuate nucleus. That's a critical region at the base of the hypothalamus that governs energy homeostasis and feeding behavior.
And what specific cells are they targeting in the arcuate nucleus?
They found direct inhibitory connections terminating on a group of cells called archegrep neurons. Archegrep neurons are the classic hunger neurons.
Okay, so when they fire, you experience a ravenous drive to find and consume calories.
Right, and during an infection, the sickness hub fires and sends an inhibitory command down the wire, forcing those hunger neurons into silence.
So aggressively turns off the drive to eat.
Exactly.
Let's do a quick final recap on this section. Bullet 1, rabies tracing shows where the neurons connect. Bullet 2, fever is caused by inhibiting the brain's natural cooling neurons. Bullet 3, appetite loss is caused by inhibiting the brain's hunger-promoting neurons.
Perfect summary.
So question to you listening, why do you think the brain uses inhibition turning things off rather than stimulation to create these sickness symptoms?
It really all comes down to energetic efficiency. Mounting an immune response generating millions of white blood cells and antibodies requires a massive caloric output.
Right.
Digestion takes energy. Thermoregulation takes energy. By simply dropping the inhibitory hammer on your digestion and letting your temperature rise by disabling the cooling system, the brain conserves enormous amounts of energy that can be entirely redirected to the immune system.
It's an energy rationing protocol.
Exactly.
So what does this all mean? Let's synthesize this entire journey. An infection starts. The immune system generates chemical alarms that drift up to the leaky OVLT border checkpoint.
Right.
The non-neuronal border guards, the neighborhood, watch detect it and blast out localized chemical sirens like IL-1 beta. The master sickness hub, our VMPOLPS neurons, hears the siren, wakes up and starts firing.
Yep.
It sends inhibitory cables down to your brainstem, effectively shutting off your biological AC to induce a fever, and silencing your hunger neurons to redirect energy to the immune fight.
It is a stunning display of biological engineering, a beautifully coordinated, centralized mechanism designed to manipulate your physical state to keep you alive.
It is deeply humbling to think about. But before we wrap up this deep dive, there is one final provocative detail buried in the paper that I want to leave you with.
Oh, the secondary projections.
Yes. When the researchers used that rabies virus to trace the wiring diagram, they didn't just find connections to the temperature and appetite centers. They found that these VMPOLPS neurons projected 12 different distinct brain areas.
That's right. And some of those target areas are heavily associated with regulating stress, processing fear, managing sleep cycles, and even controlling social withdrawal.
So consider the implications of that. We know exactly what happens when this sickness hub turns on acutely during a brief 3-day flu. But what happens if this command center gets chronically activated, say, by systemic low-grade inflammation or the long-term viral remnants we see in something like long COVID? If this hub is constantly quietly whispering inhibitory signals to your stress, fear, and sleep centers, could this exact biological pathway be the physical root cause of chronic fatigue syndrome?
It's a hugely important question.
Or even severe depression and cognitive fog that so often follows a major illness?
It is a profoundly important hypothesis. For decades, we lacked the tools to map these abstract symptoms to physical biology. Identifying this precise circuitry opens up entirely new paradigms for understanding and eventually treating chronic conditions that affect millions of people.
It proves that feeling profoundly exhausted and sick isn't just a vague feeling. It's a physical circuit in your ventral medial preoptic area actively running its program.
It really is.
Thank you for joining us on this deep dive. Stay curious, keep questioning the mechanics of the world around you, and the next time you are huddled under a blanket, sipping soup and waiting for a fever to break, take a moment to marvel at the incredible, highly orchestrated machinery firing away inside your own head.